Okay, welcome to the morning session, everybody. The last morning session of our school. How sad. However, it's my great pleasure to introduce José MEDINA-FRANCO, which you certainly know by name, one of the most prolific Chemoinformatics / medchem authors. And he has a BSc in chemistry from the National Autonomous University of Mexico, then MSc and PhD, again from the UNAM. And then in 2005, he joined the University of Arizona. There was no wall at that time. Okay. And under the supervision of Professor Majora, another big name who says neighborhood behavior says Majora. And eventually he continued to Florida at Torrey Pines Institute, then the Mayo Clinic, and eventually 2014 he returned to Mexico, UNAM. He's a full-time professor and he has his own group called DIFAQIM. So chemoinformatics, molecular modeling, artificial intelligence, application to epigenetic targets. That's the devil's work, natural products. Good luck. So he's member of the National Research System, National Science Council of Science and Technology in Mexico with the highest level. He's fellow of the Royal Society of Chemistry, sir. He was visiting professor in Montreal, University of Pereira. Maybe one day, with a bit of luck, we can add University of Strasbourg to this list. Over 250 peer-reviewed papers, 24 book chapters, 1 international patent, and okay, let's invite him to deliver his speech. And we need to press some buttons here. Where are the youngsters who know about the buttons? Well, to start the presentation. Yeah, that helps. Okay. Good. Excellent. Please, here you go.
Thank you. Well, good morning, everyone. Thank you so much for the very kind introduction. I also want to thank Professor Barnek for the kind invitation to come and participate in this event, and all the organizers and all the students. It has been a terrific conference. I like it so much. And this morning, I'm going to talk about basically an application of different chemoinformatics and molecular modeling techniques applied to identify inhibitors of epigenetic targets. So this is the outline of the talk. First, I'm going to present a very brief background of epigenetics. Then I'm going to move to the goal of our project. And then I'm going to present advances of the results in main 2 directions. One is the development of machine learning tools, and then the application of those machine learning models to identify new inhibitors of especially DNA methyltransferases and other epigenetic targets. At the end is presented a brief summary and perspectives. So first of all, I'd like to introduce my research team in UNAM in Mexico. It's called DIFAQIM. It's basically an acronym that stands for Computer-Aided Drug Design at the School of Chemistry. And similar to Professor Johannes yesterday, this is like our last pre-pandemic picture that we took actually in January 2020. And then this is on the right-hand side is our first post-pandemic picture. So very brief introduction of epigenetics. So epigenetics are modifications that happen to the chromosomes without alteration of the DNA sequence, but it still leads to a stable phenotype. There are 3 main epigenetic target families: the writers, erasers, and readers. And it's well known that alterations in histone methylation and acetylation are frequently dysregulated in cancer cells and other diseases. Historically, methylation of DNA was the first epigenetic modification linked to cancer. And as we will see in the next slide, there is a family of enzymes, the DNA methyltransferases, that are involved in this regulation of methylation of DNA. So over the past few years, we have seen, surveying in the literature, an increase in the number of peer-reviewed publications linking epigenetics with several diseases. Again, historically, the most common associated family of diseases is cancer, but in the past few years, we have seen an increase in the association between alterations of epigenetic processes with diabetes, neurodegenerative diseases, cardiovascular diseases, and some others. Also, at the bottom part of the slide, we can see also an increase in the number of peer-reviewed publications linking epigenetics with drug discovery. And slowly but surely, in the past 2-3 years, the use of the different computational techniques that we have seen in this school, chemoinformatics, molecular modeling, are of course contributing to the efforts to identify and develop epi-drugs. In this slide, I'm showing the chemical structures at the top part of 7 small molecules that are approved by the FDA for the treatment of especially different types of cancers, hitting DNA methyltransferases and HDAC. And as you can see in the picture, there are few quite natural products. Professor Johannes already mentioned about the importance of natural product research. And at the bottom part of the slide, we see examples of additional small molecules that are at different stages of clinical development. There are many more, around 30 or 40 small molecules in clinical development. And the full list, if you are interested, I would really recommend this paper of clinical epigenetics by Professor Paola Rimondo in Paris, doing a very complete survey of the current state of epidrugs. Now, as I said, one of these epigenetic modifications, DNA methylation, is basically a very simple addition of a methyl group to the carbon 5 of cytosine residues. Now, in cancer cells, it was observed an overexpression of these enzymes that regulate this process, the DNA methyltransferases, and happens frequently an hypermethylation of tumor suppressor genes. There are 3 main DNA methyltransferases. 2 of them, DNMT3A and 3B, are involved in the de novo methylation, and DNMT1 is involved in the maintenance of these methylation patterns. Now, all of the DNA methyltransferases have a regulatory domain, and most of them, except 3L, have a catalytic domain. And as you can see here, DNMT1 has this specific region that is CXXC, that, as we will see later, is kind of relevant to develop selective inhibitors towards DNMT1. So now in Protein Data Bank, there are several, like around 40 crystal structure of DNA methyltransferases for bacteria, of human DNA methyltransferases. When I started working on this project, this was back in 2007, there were no crystallographic structures of human DNMTs, but now there are many of them. The most recent one, they're quite interesting. It's this one published in September last year by GlaxoSmithKline, where in this paper, they published the results of a massive high throughput screening of internal collection of around 6 million molecules, and identified the first very selective and potent inhibitor of DNMT1. And in addition, they co-crystallized this small molecule with DNMT1. This is kind of a breakthrough because before this paper, there was no crystallographic structure of DNMT1 bound to a small molecule. And as you can see in the slide, this small molecule has an IC50 for DNMT1 of around 36 nanomolar, and basically is not inhibiting DNMT3A and 3B. Now, this slide shows examples of small molecules that are either non-inhibitors of DNA methyltransferases or have reported the capacity to demethylate the DNA in cells. I like to point out that this couple of cytosine analogs, 5-azacitidine and 5-aza-deoxycytidine, these two actually are drugs approved by the FDA for the treatment of myelodysplastic syndrome. But as you can see from the chemical structure, since they are cytosine analogs, the mechanism of action is that actually they bound to the DNA, are actually prodrugs, are quite potent, but are also quite toxic. So this is why the scientific community, several other groups in the world, including our group, are trying to identify other small molecules, but with very different scaffold. There are several natural products reported in the literature. In the slide, are just a couple of them. Other groups are working on repurposing, drug repurposing. All these techniques that we have seen in this wonder summer school have led to very interesting chemical scaffold doing virtual screening. Also, classical medicinal chemistry efforts, synthesis, have contributed to additional potent compounds. And as I just mentioned before, additional small molecules have been identified doing high throughput screening of very large chemical libraries. But with this very brief background of epigenetics, now I can tell you that the goal of our research group in this area is to discover and develop DNA methyltransferase inhibitors and other inhibitors for other epigenetic targets using combination of computational approaches with experimental validation. Now, in the next few slides, I'm going to present progress towards this goal, basically addressing two specific aims. Models in virtual screening of compound libraries. Just probably is kind of a fancy name. We are using this term of epi-informatics. This actually was a book that I had the chance to edit following up an ACS meeting in San Francisco in 2014. And this is basically like the use of these computational approaches towards epigenetic drug discovery. Anyway, as you can see, I don't need to tell you that as an academic group, our major source of SAR are public databases like these two. And the first step, of course, was to extract the SAR published for small molecules targeting epigenetic drugs, epigenetic targets, excuse me, from Campbell. This table is a screenshot of a partial list of the SAR published in Campbell of small molecules targeting more than 50 epigenetic targets. As we can see from this slide, there are epigenetic targets like HDACs, histone deacetylases that have been heavily studied by industry, academic groups. At the time of that work, the full report was published in 2008. For HDAC1, there were more than 3,000 compounds. There are other epigenetic targets that have much fewer data points. Here are our DNA methyltransferases. At the time, there was data for 248 small molecules. Now, in the latest version, there is information for more than 500, at least in Campbell. So based on the data from Campbell, Norberto Sanchez, former PhD student in the group, developed predictive models using support vector machines. He tested different representations. In this case, I'm showing the results of extended connectivity fingerprints 4 and RDKit fingerprints, also a consensus model, trying to improve the predictive ability. And overall, just to make the story extremely short, the models developed have a very decent binary classification and target prediction power. The full details of these models are reported in JMedChem 2021. Now, the second step was to make these models available especially for the medicinal chemists working in the field. So again, Norberto Sanchez, part of his PhD thesis, developed this web server. It's publicly available. It's called Epigenetic Target Profiler. It's basically the implementation of these models, but in a much more user-friendly. So as many other servers that we have seen, and for instance, there was a fantastic poster, the Plateau, for predicting targets. This is focused on epigenetic targets. The user basically draws the chemical structures or input the deciles, and the server is gonna produce this type of table, the results. Here is the input structure. Then on the right-hand side is the process structure. I mean, the server is doing an internal standardization. And then at the bottom is displayed the list of epigenetic targets. Of course, these are predictions. And in the last column here, the quartile is a measure of kind of the confidence of the predictions. So the higher the quartile means that that pairing molecule is more similar to is more within the applicability domain of the predictive models. So right after we made public this server on LinkedIn and social media, we have this kind of encouraging comment from Professor Ursu at the Scripps Research Institute in Florida, that his group, as you can read, tested the server with the compounds. We don't know what compounds they are working on, but at least the server gave a good prediction for these compounds. Of course, this is not the ultimate proof, but at least it's kind of palm on the back that it's been helpful. And of course, we are keep using this server for internal purposes, and everybody's free to use. So the implementation of the web application itself is reported here in JCIM, also in 2001. Again, work of Norberto Sanchez. Now, I just presented extremely briefly like the efforts to develop these predictive models. Now, in this second part, we're going to see the use of those models to identify inhibitors of DNMT1s. We're following a very classic virtual screening approach. At this point, nothing fancy. So in terms of the compound libraries that we're exploring, we're interested in compounds from different nature. One of them is natural products, also food chemicals, in-house data sets from collaborators, commercial synthetic compounds, and more recently, and I will show these results later, focused libraries on epigenetic targets. For the computational, the filtering part, we're using combination of similarity searching, molecular docking, dynamics, and the use of the machine learning models that I just briefly introduced. For the experimental validation, the first step that at least we're implementing is basic, but I think important enzymatic inhibition assays, and try to collaborate with other groups to test these compounds in vivo systems. That is more complicated. For the optimization part, again, kind of classical medchem lead optimization, and this is done in collaboration with medicinal chemists in Mexico and in other countries, and as I will show. So a few more words about the compound libraries. These are the natural product libraries. Also in this conference, we have seen the use of coconut. We are also searching natural products from Mexico. We collected in a separate effort a database of around 530 natural products, isolated and identified. In Mexico, there are many groups working on the natural products area. So basically, we're putting these compounds together in a database that is also publicly available. And also quite interested in food chemicals. And this is because also surveying in the literature, there are many food components that, of course, are not used for therapeutical purposes, but regulate our epigenetic processes in a natural way. So here, the rationale behind this is to rationally or at least systematically identified food components that are targeting our epigenetic targets that we have. And in this regard, again, this is not for therapeutical purposes, but it's kind of envisioned to like for more disease prevention in that regard. Now, data set from collaborators, just a couple of examples. Dr. Laura Alvarez and Mayra Antunes are working on natural product derivatives. They are based in Morelos. It's a beautiful state, very close to Mexico City. And also the group, Professor Alexander Gagnon in Montreal, Canada, is also interested in developing epigenetic modifiers. His group is focused on medicinal chemistry, so we're working together to combine efforts, basically. Commercial libraries, basically from zinc, and also in the past 2-3 years, we've seen that several chemical companies are assembling focused libraries on epigenetic targets. So here the idea is, in addition to screen other types of libraries, identify the potential inhibitors in these kind of pre-selected enriched libraries to see if we increase the chance to find active molecules. And as I said, our group started working on this project several years ago. This was the first report back in 2010. As you can see, here again, we implemented a very classic standard virtual screening workflow, starting from a public database of the NCI compounds, 260,000, then select lead-like molecules, and then implement a sequential or cascade docking protocol, combining, in this case, glide high-throughput virtual screening, then glide standard precision, and then use 3 different docking algorithms, GlideXP, GOLD, and AutoDock, to select consensus hits from the initial pool of 260,000 molecules, ended up with 24 consensus hits, out of which 15 were physically available for experimental assays. So the 15 compounds were sent directly from the NCI in Washington to our collaborators at the time, the group of Dr. Frank Laiko at the German Cancer Center in Heidelberg, and they did the enzymatic inhibition assays. So these are the chemical structures of 7 of the 15 compounds that they tested that show some degree of enzymatic inhibition towards DNMT1. So the potency of these compounds was not that significant, was not like aha moment, these are the best molecules. But what was relevant is that the chemical scaffolds of the structures were quite different from the scaffolds that at the time were published for DNMT1. So back in 2010, in the literature, there were about 20 compounds in the literature. Now, as I said, there are more than 500. One of the hits from that, I would say, basically like exercise, was a natural product, nanomycin. Is an antibiotic, but as you can see, it turned out to be potent and selective. This is nice. Selective towards DNMT3B. Showed an inhibition of 500 nanomolar and did not inhibit DNMT1. And at this moment, so again, this is back from 2010, so far there are no other small molecule that is selective towards DNMT3B. This compound not only showed inhibition in enzymatic biochemical assays, but also showed the methylating activity in cancer cell lines. Now, going back to our list of the hit compounds, going back to the chemical scaffolds, over the next few years, our group and other groups use these chemical scaffolds to initiate this hit-to-lead optimization program. So when I was at Mayo Clinic, we collaborate with Professor Nathalie Maurice, and doing a virtual screening, a shape-based similarity searching of a drug bank, identify or salicin, as you can see, is extremely similar to our query compound, also turned out to be quite potent inhibitor of DNMT1. Independently, the group of Professor Alexander Gagnon, at the time we didn't know each other, he just took the publication and his group decided to take that chemical scaffold and doing this medchem optimization process, increase the potency of, make series of analogs and increase the potency of that chemical scaffold. Also, in an independent work, Professor Massimo Bertinaria at the University of Turin started from that chemical scaffold and also optimized the activity. And more recently, the group of Yuan Xiao in China also go back to the same chemical scaffold of Professor Alexander Gagnon, and his group developed dual inhibitors of DNMT1 and HDAC inhibitors. This is kind of towards the polypharmacology, but in epigenetics. Now we have continued over the years to do virtual screening of different chemical libraries. This is just kind of a snapshot of some of the results. We have identified additional approved drugs, in this case, glyburide and panavinostat, and as you know, are approved drugs for diabetes. So they also have some degree of activity towards DNMT1. Additional, one natural product, dafablin, that is found in black tea, and additional small molecules from commercial libraries. So right now, the group of Professor Federica Cati at Arkansas State University is working on that chemical scaffold to optimize the activity of this compound. This project is working one of the students in UNAM, Diana Prado, that right now is visiting the University of Barcelona. And this is nice for Diana because now he's doing her PhD, is doing the structure-based design, but also has experience in medicinal chemistry. She did his master's in chemical synthesis. Now she's kind of experiencing both, a little bit of hands-on in the lab to test how difficult I'll actually make the molecules that she is predicting. So I think for her it's quite good. Anyway, here in the next few slides, I'm going to show briefly the efforts towards first the characterization of focused libraries on epigenetics, and then the use of those libraries to identify additional hit compounds. So the first step in this, let's say, project was to collect, of course, systematically the commercial epigenetic focused libraries. This is a work of Alexis Padilla. The table shows a summary of the different chemical companies that have out there these focused libraries and the number of molecules before and after data curation. Of course, the chemical companies are going to say that the compounds are drug-like, are very nice, are perfect, but kind of a sanity check is always, as Professor Topsha says, like trust, but verify. So verify that they are really drug-like. So this is, as you can, is quite simple, quite fast. Explore the drug likeness properties of these chemical libraries. This is just as an example, a visualization of chemical space using standard PCA. Each focused library is identified from different colors, and the take-home message of this analysis is that, well, yes, indeed, these compounds are kind of drug-like, but from this type of analysis, we see a diversity among these libraries. Now, also part of the exploration of the focused libraries, we implemented additional chemical space analysis. This is another example showing what is called Constellation Map. This is another way to visualize chemical space, and I'm not sure if you are familiar with these constellation maps. So very briefly, if you're not, each data point in a constellation maps represent not only a single compound, but represents a series of analogs. So that concept is is based on the work of Professor Bajorat of analog series. So the size of the data point depends on the number of compounds that are populating that analog series. So of course, the larger data point, the more compounds are in that analog series. And the edges that connect some of these analog series are common chemical scaffolds. So the analog series that are chemically related forms, maybe again, just a fancy name, just make this analogy, forms a constellation in the chemical space. Also following up this analogy, also a little bit for fun, we are calling here this chemical scaffold or polar star, meaning that is kind of the most representative chemical structure among this universe of epigenetic libraries. Kind of a reference structure. So this analysis is extremely fast to do. The full details of constellation maps that has been used in several other projects are published in this paper, Frontiers in 2019. Now, in these constellation maps, we can color code each individual epigenetic library just using a color. For instance, here in the second panel, we see how the library from chemical diversity is covering a wide region of the chemical space, and there are other focused libraries that are much less diverse. However, for instance, this from Acinex. From Acinex here, it's much more, it's less diverse, but is occupying a region of chemical space that is not occupying chemical diversity. And this type of comparisons can easily visually done in these constellation maps. Also part of the characterization of these epigenetic libraries, we implemented this as a collaboration with the group of Ramon Miranda. He's working at the University of Florida. He developed the chemical library networks. That is a way to visualize the chemical space of quite large libraries, like for instance, in this publication, with him, we developed this, we'll use these chemical library networks to represent the chemical space even of the entire zinc library. It's going to look like this. This is actually a chemical library network for epigenetic focused libraries. And ChemDiff, again, the most representative chemical library here is the node number 2. That is kind of at the center of the chemical space. Now, once we have kind of analyzed the libraries, comes the virtual screening. So this is a summary of the protocol to do the virtual screening. It's basically a combination of machine learning models, consensus docking using 3 different docking programs, MOE, Autodoc MEDINA, and Autodoc, and then rescoring the docking scores using extended connectivity interaction features. Also, this is a recent method developed by Norberto Sanchez. The full details are published here in Bioinformatics 2021. So going back to the initial pool of compounds, more than 53,000, as you know, applying these machine learning models in virtual screening is extremely fast. So here we selected 119 compounds with the best predictions. Then this number of compounds were subject to the consensus docking and rescoring, and finally select 20 compounds for purchase and enzymatic inhibition assays. This is a partial list of the 20 compounds, just showing the docking scores and the rescoring with this protocol, ESIF. Now we send those compounds to Reaction in Biology, a chemical company that performs the enzymatic assay. This is in Pennsylvania. And out of the 20 compounds, these quinolines in this slide showed some degree of inhibition between 10 and 40%. This is at one single concentration. And here it was kind of interesting to observe an activity cliff. This is kind of a textbook activity cliff. You can see, the chemical structures of these molecules are basically identical except for these 2 methyl groups. However, if there are 2 methyl groups in the chemical structure, it's kind of a decent inhibitor, around 40%. But if we don't have those methyl groups, it's actually an activator of DNMT1. So we are still kind of wondering now to rationalize why this is an activity cliff. Now, this is the most interesting result. From that experimental screening, we obtained 2 quinazolines, shown here in the slide, that had an IC50 towards DNMT1, one of them 30 nanomolar. It's kind of comparable to the activity of the GlaxoSmithKline that obtained doing that massive high-throughput screening of 6 million compounds. The degree of activity is kind of similar. And analog, as you can see from the chemical structure, just here as a small difference, showed an inhibition of DNMT1 of 81 nanomolar. Both compounds were much less active towards DNMT3A and were not active towards DNMT3B. Just for comparison, as you know, we have an internal and positive control. The reference standard control is SAH that has an IC50 of 0.34 micromolar. This is actually a natural autoinhibitor. So these compounds are looking quite promising. The results were just kind of new, just published earlier this year. Now, surveying in the literature turns out to be that both quinazolines are also inhibitors of a second epigenetic target, an epigenetic target reader, G9A. And from this paper published in JMedChem, for a group in North Carolina, one of those compounds has an IC50 for J9A of 6 nanomolar. So putting together our results with the published results, we can find out that these quinazolines are dual inhibitors of 2 epigenetic targets. So really promising candidates for polypharmacology. So trying to figure out the mechanism of inhibition, this is just very briefly an effort to see why these compounds are selective. This is results of Edgar Lopez, now a PhD student. He did classical docking molecular dynamics to see where this compound might be binding, trying to rationalize the selectivity. And again, to make the story extremely short, the end result was that it seems that these quinazolines are actually interacting with the CXX C domain that, as I mentioned before, is not present in DNMT3A and 3B. So partially this kind of explains or gives us a hypothesis why this compound might be selective. Also from the experimental side, of course, also in collaboration with reaction biology, they did experimental competition assays with DNA. So that is kind of the binding site of the catalytic site, and the results confirm that these compounds are not binding in the catalytic site. So it seems that, at least kind of makes sense, the molecular modeling results with the experimental results. Of course, we need to do additional experimental and computational assays. Just to conclude, brief summary. I just here quickly presented the identification of DNM inhibitors that, as I mentioned, has been starting points for hit-to-lead optimization programs. Mentioned nanomycin is the first and so far the only selective inhibitor for DNMT3B. And I just also talk about a couple of quinazolines that are potent inhibitors of DNMT1 and are also inhibitors of G9A. So, and as everybody in this room knows, the SAR published in the literature has been quite useful to develop machine learning models. In this specific case, I talk about epigenetic target profiler that is quite useful to actually use those models to identify additional compounds. Perspectives in our group in this field is use machine learning models to identify systematically dual inhibitors of the DNMT1 and also other epigenetic targets. So from the literature, we know that there are several compounds with dual inhibition of DNMT1 and HDAC. For instance, the example that I showed you before, the group Dr. Yao in China. Now the idea is to do that prediction, but in a systematic way. So second perspective is to implement the novel design techniques that has been talked about a lot in this summer school. Interestingly, the novel design has not been used, at least published, for DNMT, as far as we know. So we're using all these techniques that we have been learning in this summer school in this effort. So just to conclude, I'd like to acknowledge again current and former members of the FAKIM group, collaborators that kind of make reality or dreaming predictions with computers. Also, many thanks to the funding agencies. Many thanks to you for your attention. I'd like to briefly show some pictures of UNAM in Mexico City. This is the School of Chemistry that, because of the 2011 periodic year table, so they display the periodic table in the building. This is the main library, the School of Medicine that is next to the School of Chemistry. Of course, we have the soccer stadium, home of the Pumas, and the concert hall. Many thank you for your attention.
Well, excellent talk. Thank you a lot. Questions?
Thank you very much for a wonderful talk. So the compounds you found are the molar ones. So basically they are known GNI8 inhibitors, and Athos Therapeutics owns the patent which covers absolutely everything around that scaffold, I promise. So do you think that patent will be a problem for you to work on DNMT1?
I think so, yeah. So, I mean, now based on these results, also maybe one of the next steps is to actually talk to the group and see if they have been working by any chance also with DNMT1 and try to join effort. It's not basically kind of a competition, but trying to see if we can work together to further develop these compounds.
I also have interest in these compounds.
Thank you.
Okay, any more?
So first of all, thank you for the great talk. My question is the selectivity to DNMT1 when it's selective. So when it inhibit the DNMT1, would it be also inhibited in the normal cells or just would be in the cancer cells? Because the cytotoxicity of the cell wouldn't be avoided, I guess.
Yeah, well, that's a great question, and of course that's kind of the next step, to test these compounds in actually cancer cells and see if an in vivo scenario are able to demethylate DNA. Now, from that other paper, that other group in North Carolina has published a lot. Those compounds are active in cancer cells, but at least the publication is focused on the activity of GNA. So now from this point of view, yeah, our next step is to actually find a collaborator, because it's sometimes hard to find someone to, yeah, but that's our next step.
Okay, well, we are 3 minutes late. Let's thank our speaker again.